So let's get started. So basically, we'll discuss the contents of week three. I'll walk you over some algorithms and how to implement those algorithms. And after that, as usual, we'll solve a few MCQ questions. And yeah, so let's get started. So uh, the first topic for today is assortativity of networks. So this was a topic that has been discussed quite well by Karthik sir, but I'll still cover it a little bit. So basically, what do you mean by assortativity? In layman's term, it is basically uh, the property of the nodes to connect to other nodes within similar with similar properties within the network. So basically, if you imagine a transcription regulatory network, the similar kind of nodes, say for example, genes are connected to genes only, or proteins are connected to proteins or transcription factors. That would be wrong because transcription factors would be connected to genes, but you get my point. The thing is, similar kind of nodes will connect to each other, having in the case of biological conditions, you can imagine them having similar kind of functions. So that is assortativity, and the opposite of uh, which is basically disortativity. Basically, the property of the nodes to connect to other nodes with dissimilar properties. So the point is, basically the degrees are a metric measured on individual nodes, but when we focus on edges, there are always 2 degrees associated with each edge, one for the node where the edge originates and one for the node to where the edge points. So basically, if we take out these two variables and we uh, plot them on a scatter plot and fit a line through it, we get a correlation coefficient. So basically, if your network is assortative in nature, the if you plot the uh, basically the edges between similar nodes, it should have high correlation. Not one, but it is close to one. So yeah, and the mathematical formulation of assortativity is given by this formula, where here E is basically uh, E is basically a matrix. It represents the fraction of edges in the network between I and J. So uh, EIJ is a matrix that uh, gives you the fraction of edges between network node type I and node type J. A fraction in the sense like you get the total number of edges and divided by the total number yeah, for each ij pair. So that's how you get the fraction of edges. And basically, the trace of the matrix is basically it's a linear algebra concept. It's the sum of the main diagonal, so leading diagonal elements. If you sum that up, that's the trace of the matrix. And it is, and if you the formula of assortativity is given by trace of E minus mod of E squared by 1 minus E squared. So we'll not go into the mathematical concepts, mathematical formulations. More will focus on the concepts and the applications and how they relate to our studies in system biology, network biology, etc. Okay, so moving on. Next is the centrality lethality hypothesis. So it is a continuation of studying the assortativity of a network only. And basically, the hypothesis states that here I've written proteins, but you can imagine as any nodes that can be genes or that can be proteins or transcription factors or whatever metabolites. So basically, proteins with a higher degree centrality is more likely to be lethal, and proteins involved basically, which means that proteins involved in more interactions are more likely to cause death when knocked off. Basically, you can imagine it as a if you imagine a signaling cascade. So if your node has a very high centrality, basically it is it is connected to a lot of nodes and within your network. So it implies that that node is very essential to your network. It is basically part of many you can say processes or signaling pathways or metabolic reactions. It is part of many such modules, and it is regulating all of them. And so if you knock that node out, or particularly that gene or that protein out, it is going to cause you the greatest harm. So basically, compared to another node which has less centrality in your network, if you knock that out, it is more likely that that node is not involved in like many processes as a node which has higher centrality. So basically, here what they are trying to say is they are implying that centrality, uh, increase in centrality of a node is equivalent to, it's basically associated with increase in lethality of that node. Here, lethality means basically the importance of the node in maintaining the regulatory system of the cellular system. So it basically tells you how important a node is. So this proposition has given rise to several new investigations in which stronger associations were obtained for other centrality measures as well, not only degree centrality. There can be, like, you can imagine that as betweenness centrality as well. So if your node has a higher betweenness centrality, it means it is kind of connecting bridges between a lot of modules. So it is involved. It is basically shared across a lot of modules. It is connected. So uh, yeah, so higher your edge betweenness, higher will be your impact of that node in the network. So yeah, not only degree centrality, other centrality measures can also be basically uh, applied in this case. So yeah, most of the previous work on this basically focused on well-known protein interaction network of Saccharomyces cerevisiae. Uh, in a recent study, however, it was found that the degree and the betweenness of lethal proteins is significantly high, above average across 20 different protein-protein networks. So, and closeness centrality on the other end did not perform well. So, yeah, so basically, not all the centrality measures should be following the centrality lethality hypothesis. For example, here the closeness centrality is not followed, but degree centrality and betweenness centrality, as like you can think of from your intuition, will follow the centrality lethality hypothesis. There's how do you define how central a node is? How do you define how important that node is to your network? So here is a metric that we use to define that. It is called the pairwise disconnectivity index. So basically, this index quantifies how crucial an individual element is for sustaining the communication ability between connected pairs of vertices in a network that is displayed as a directed graph. Such an element might be a vertex, which in our case can be molecules or genes, or it can be edges, which can represent interactions or reactions, or actually group of edges or a group of nodes. So you can think them of a gene set or a set of reactions uh, like that. And the index can be viewed as a measure of topological redundancy of regulatory paths, which connect different parts of a given network as a measure of sensitivity of the network. Basically, uh, how do you measure sensitivity or robustness of a network? So basically, you try to knock out certain nodes, you try to knock out certain edges and see if the network performs as before. But if your network is robust enough, it will not be perturbed by it. Yeah, I mean, technically it will be, but in the whole sense, in the whole communication sense, or the whole information passing or information diffusion sense, the network will not be much affected if you have a lot of uh, basically if your robustness of the network or the robustness is high. Basically, robustness and sensitivity are kind of the opposite. So if it is getting perturbed very easily, you can say it is a very sensitive network. On the other hand, if it is not getting perturbed very easily, you can call it a robust network, just to keep this in mind. And uh, pairwise disconnectivity index is given by this form where n naught minus nv by n naught. So here the n naught is the total number of vertex pairs in the network that are connected by a path of any length in the network. So if like they can be uh, like two opposite ends of a network, they can be on the diameter of the network, and the nv as in like the n naught has no specific like basically like no metric involved in it. N naught is calculated between every pairs of vertices in the network that are connected by you have to see if A and B nodes are connected. If connected, then you calculate the N naught. Uh, and NV is the number of vertex pairs that remain connected following the removal of vertex V. So you take the network for every two nodes, you calculate if there is a path between the two nodes, and that that N naught is the number of that paths, and NV is the number of vertex pairs when you remove that node. So you remove that node and you see if there are any if left paths that connect these two nodes. So that is your NV. So this is given as a fraction. So higher your disconnectivity index, it means like the less involved it is in the network, and less um, lethal it is to your network. Next, this is a diagram of a Saccharomyces cerevisiae protein-protein interaction network. So this image has been shown in in the original videos as well. Sir talked about this. So basically, this is uh, pro- a protein-protein interaction network of S cerevisiae, and the nodes are colored according to how they are important, how much importance they hold to the network. So the red ones are the lethal ones, the green ones are the non-lethal, the orange and yellow are basically not characterized and uh, like not much involved in the network, right, in terms of centrality or lethality. So as you can see, all the leaves of these networks, a network on I'm talking about the nodes on the edges of the network, those are mostly green, and the nodes that are within like in the central position of the network, you can see that those are mostly red. So basically, all the lethal proteins that are involved in this protein-protein interaction network of S. cerevisiae, they are concentrated mostly within the center of the network, which means they are they have more degree centrality compared to the nodes that are present on the like surface of the network away from the central positions. So the lethal proteins are more connected, and which in turn intuitively helps you understand that it is more involved in different processes. So this kind of holds up our centrality lethality hypothesis that we talked about. A node which is situated more centrally and more connected, that is more lethal if knocked out. So yeah, so the red ones are more centrally connected and they are more lethal when they're knocked out. And these two graphs are given as basically this you can imagine as the left one as a degree distribution, and the right one is basically a graph between percentage of essential proteins and the number of links. So higher percentage of close to like if you see at these points 60 to 80 ages, these proteins, if the last part basically 60 to 80 range, these proteins 60 to 80% of this protein of the proteins have kind of 15 to 20 edges, 15 to 20 links you can say. So basically essential proteins which are uh, important for the cell to grow, for the cell to divide, and perform its various metabolic and cellular activities. The essential proteins are more involved in different modules or different signaling pathways or different reactions. So that is a kind of the outcome of this of the analysis they did. So basically, this is kind of in line of with the centrality lethality hypothesis that we talked about. Say, saying the essential proteins which are more involved in the network will have higher number of links. Excuse me. Yeah. So moving on. Next is basically network perturbations in biological networks. So interactions among molecule cells and individuals determine the properties and behavior of a biological system. These interactions give rise to various biological networks and their mathematical representations. And we can use these biological networks for integrating different data types. So basically, we can use different kinds of, in most cases, we use omics data to integrate into these kind of networks uh, uh, basically where you can where you can understand in particular cases, for example, in disease networks, where you can understand how different mutations or how different uh, like basically knock any kind of perturbation to a disease to a disease network can uh, can help from that disease. So basically, you have a network. For example, if you consider these networks, so basically this is a healthy gene regulatory network. So if you see this pathway, so this transcription factor influences three genes: gene one, gene two, gene three. Again, gene three produces a transcription factor which influences gene four, which forms a feedback loop to transcription factor one. So what will be the disease form of this network. So basically, in the diseased form, the fourth gene that is being controlled by the transcription factor 4, this is mutated. So basically what happens is this regulation is not done properly because of this mutation in the G4 gene, mutated gene. So this regulation is not done properly, so which affects this regulation, which is basically indicates that this red dot in this transcription factor 1 is basically the site at which this is basically the site at which it binds to the gene 1 and regulates its activity. But since this binding, basically, since um, you have a mutation here, this binding or this regulation is not done properly. So this active state is not open for gene 1 to bind to the transcription factor 1. So what happens is this whole regulation is lost. So the gene 1 regulation involving transcription factor 2 and a further gene downstream, this whole network is lost because of just a mutation on a gene that is forming a feedback loop on the original transcription factor. So basically, and these molecular changes, such as mutations, can be regarded as perturbations of a biological network, and the connectivity of the network is used to predict the effect of the perturbation on the system. So basically, you can predict from networks like these and using certain models that how these genes and transcription factors are going to interact if some of them are mutated or a few of them or like even a single one of them is mutated. So these kind of assumptions and these kind of predictions can be made through these networks. We'll look into details, more details about gene regulatory network. But yeah, uh, the connectivity of the network can be used to predict the effect of the perturbation of the system. So if it is more connected, that means you're in greater trouble. That means more of the downstream proteins or genes will be affected, leading to right, a more drastic condition. So you can how you can do this analysis is you can employ graph diffusion techniques to estimate the system level effect of individual patterns and perturbations, and how these lead to differential expression of genes in tumor cells. In this case, it's tumor cells. In yeah, other cases, it will be in that particular disease. Just a second. Somebody is okay. Okay. So moving on, we'll talk about some examples where diseases can be like thought of as network perturbations, like certain nodes in that network, be it genes or RNAs or metabolites, they have been perturbed in the network that has led to the formation of diseases. So we'll see a few examples of it. Uh, 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 basically, yeah. In the next from the next slide, we'll see some examples of that. But yeah, so how can you imagine disease as network perturbations is basically uh, the assumption is that cellular networks are very robust in nature, that small perturbations or small changes in the network do not bring about changes in the whole network topology or the whole network uh, in terms of information flow or diffusion, information diffusion. So basically, your assumption is that cellular networks are robust, and but we'll see also in the next few slides, there are specific locations in the networks known as hubs or places where the nodes are more densely connected than other places in the network. So if you target those hubs, uh, uh, the 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 robustness of the network can fall very easily. They can be very fragile to those perturbations, in particular hubs or particular important nodes in the network. So you can think of it as those nodes only which are more centrally connected. So those kind of make up the hubs in the network. We'll talk about a little bit about those in the community direction and modified identification slides. But the point is, yeah, if a node is centrally located and highly connected, it is possible that it forms a hub with certain other nodes in near near it in the network. So these hubs are very sensitive to perturbations. The whole network might be robust, but if you target specific hubs in the network, those are sensitive to those perturbations. And this a trade-off between this robustness and fragility can lead to this disease formation. So certain gene mutations or certain combinations of gene mutations, not even one, like one gene mutation like may falter its downstream processes. But if you have multiple genes that are mutated and which act in tandem with each other, if those are mutated together, you can have a different kind of phenotype altogether. So these can cause this can trigger cascades of failure in gene regulatory networks, which can lead to disease phenotypes. And examples of such diseases are Huntington's disease, cystic fibrosis, sickle cell anemia. Uh, uh, Basically, these are monogenic diseases. What by monogenic uh, here means is basically it is caused by a mutation in a single gene. Whereas other diseases like cancer, diabetes, and Alzheimer's, these are like complex multifactorial diseases. You cannot pinpoint the reason to a mutation to a particular gene. So there are various factors involved, various genes involved in this in bringing out phenotypes like cancers and Alzheimer's. So these modifiers genes also, there are modifier genes as well, and there are environmental factors as well. So basically you can have genes which are not involved in protein synthesis directly, or genes that regulate protein synthesis but not directly. So basically you can have genes like miRNA or certain other kinds of genes which can play a role in the disease formation. Basically, mutations or like incorrect bindings or knockdowns can basically, if you think of it, miRNA as an example, miRNA is basically used to knock down a lot of protein coding mRNA in the cell. So basically, the protein coding mRNA will be single stranded, and if you have an miRNA, it goes and directly binds to that single stranded mRNA, making it double stranded. And once it is double stranded, it cannot be it cannot undergo translation. So you kind of you can knock down certain genes in the system using miRNAs and similar other genes available as well. So these kind of genes and also environmental factors so if you think of cancer, uh, environmental carcinogens like UV light can have a big say in the disease manifestation and progression. So this can influence gene interactions and pathways, which can lead to disease formations. Okay, moving on, we'll see some examples, 4 or 5 examples of how network perturbations in systems biology level approaches are used to study certain diseases. Uh, okay, so the first example is basically a study where they examined the dynamic structure of the human protein-protein interaction network to predict breast cancer outcomes. So what they did was they computationally removed the nodes from the PPI, critical protein interaction hubs, the hubs that I talked about, like the proteins that are situated more centrally in the network and more connected in the network, having a greater say on how the the cellular processes work. So basically, you can computationally remove these nodes using programs like NetworkX or in MATLAB, and you can remove these critical protein interaction hubs as well, and see how the network connectivity changes and how the information flow changes. So basically, uh, the hubs are classified into two types: the intermodular and uh,
Excuse me. Yeah. So, uh,
yeah.
so on what basis are like the critical protein interaction hubs identified? As in, uh,
So
is it
mm,
purely based on the like network properties, or is it based on some other factors?
okay, so we'll see in some slides down the line. So there are communities that are getting formed in the network. So
Yeah.
once you identify those communities, you can see certain nodes within those communities which have a higher, say, degree, basically higher in degree or out degree in case of directed graphs. So also, not only is the connection important, the another important factor is the function of that node. So
Yeah. Yeah.
not only in terms of like in terms of like hubs are getting formed, not in only terms of connection, also how important is the function of that node to the whole module. So if you imagine a whole protein-protein interaction network, there can be modules formed of, say for example, one module is getting formed of glycolysis, one module is
Yeah.
getting formed of Krebs cycle, one module is getting formed of, say, uh, electron transport chain, whatever.
Mm,
So
yeah.
so the the protein the basically the node within the glycolysis module, which will have higher connectivity and which will be functionally more relevant to the cycle, will be treated as basically the hub protein. And here I'm talking about a single hub protein. You can have multiple hub proteins, so you can
Yeah. Yeah.
have a whole community of hub proteins within a certain
Yeah.
module that is regulating that function of that module. So not only in terms of degrees,
Yeah.
you have to think
No,
in terms of functions
yeah.
as well.
Yeah, yeah. But I was wondering, in conditions where suppose there are certain proteins that are like we don't totally understand the function, but we have identified these proteins. So
Hmm.
say in these conditions where we have found higher degree, so like still that will be considered as a critical like or hub protein.
If you do not know the function, you can assume it to be a hub. But as I was saying, like if you have to be basically if you want to define a node or a group of node as a hub, so you have to say that those nodes are very critical to that module. So
Yeah, yeah, yeah,
critically, you can define it terms of degrees, but it is always favorable to define in terms
yeah,
of function. So only degree saying okay,
yeah,
that this node has a higher degree has, that means this is a hub. Even if you don't know the function, that would be
yeah.
kind of factually wrong.
Yeah.
So
No,
you
but
can
I'm just saying the condition where we don't know the function or like somebody like
very
we are
yeah.
assuming that it could potentially be like have a more relevant function or something like that.
So then you can develop models where you
Yeah.
assume that this is a
Okay,
hub,
this is okay.
and you create a null model where you assume that
Mm,
this is in the hub, and you see how the network's properties are
okay.
changing according to your model.
Okay.
So that you can do. But in general, like if you want to characterize a protein or a node or a group of
The
nodes as
function.
hubs, the function is important.
Okay.
Okay. Okay. Yeah. So basically, mm, y- this study basically studied the dynamic interaction of the protein-protein interaction networks, and they identified hubs within those networks which are critical to breast cancer. So and they found that these intermodular hubs, basically hubs connecting two modules, are quite crucial for this connectivity, and also they were associated with cancer phenotypes more than the intramodular hubs. So basically, what they're claiming is the hubs that are connecting two modules are more important than the hubs that are present within the modules. So this is a claim, but this is not completely true, but it is true in this study. Just a second, somebody is trying to enter. Okay, and the next study I talked about is basically the miRNAs that I talked about. So they compared the miRNAs in the regulatory networks in normal tissues and compared it to like how they acted in various cancer tissues, kind of your control and uh, cancer yeah, kind of your how you do your control and cancer sampling. So you have a network where the miRNAs, if you have a network in a normal cell and you have a network in a cancer cell, and you compare how the miRNAs are connected and how the information is passing on from in a cancer cell, in a cancer network compared to a healthy or a normal network. So what they found is normal tissues exhibited complete miRNA networks. Cancer tissues were represented by disconnected subnetworks. Basically, the miRNA networks were not complete and they were not connected well enough. So basically, they identified certain miRNA cliques in those networks with which they could associate with certain cancer types. Next study was basically a study where they constructed a functional protein interaction network using different sources to analyze two data sets from the. This is basically a consortium or a multiform or basically a data set that is already available. So it's basically glioblastoma. It's a cancer of the nervous system, and they mapped out. Yeah, they basically constructed this protein interaction network. They did not have this. They constructed it from the data set that they have, and they mapped out cancer candidate genes into the network, and and it revealed that the clustering in those modules enriched with known oncogenes, tumor suppressors, and genes involved in signal transduction, suggesting the presence of a core network in GBM tumorigenesis. So they constructed a GBM network from the data that they have. I'm hoping that's kind of a mixed data only. So they constructed this network, and they mapped out certain genes that they already knew were involved in cancer, and they found that those genes kind of clustered with with previously known oncogenes and genes that were involved in signal transduction. So they basically identified a core network within the whole network, where you can say that that was the hub for the GBM tumorigenesis. Another study is basically where they uh, kind of combined the study of human metabolic network topology and skeletal muscle gene expression dataset, and they were trying to identify uh, signatures of signatures or proteins and molecules involved in type 2 diabetes. So they basically pinpointed potential drug targets and clinical diagnosis, including transcription factor and metabolites. Basically, how they did this, I have not explained it here. So you can do this, like how you can identify certain drug targets is also through the thing that we are discussing. That is basically, if you want to target a drug for a particular disease, you would want to target it to a more influential protein, a protein or a protein, a set of proteins which are more influential in that phenotype compared to other proteins. So basically, you want to target a hub within a network for a particular uh, drug that you want to target for a disease. So here also the importance of hubs and the functions of those hubs come into play. So when you're trying to create a drug for a particular disease. So yeah, they did some perturbation analysis on the cellular metabolic network and identified highly connected metabolites, including NAD plus and ATP, and they identified them as contributors to widespread gene expression changes observed in type 2 diabetes, which is like if you think of it now, it's kind of intuitive because these are involved in metabolic processes and basically glycolysis and gluconeogenesis. So this should be involved in type 2 diabetes. But this was kind of a proof that came from a network point of view. Another one was the study on initiation and progression of murine prion disease. Murine is basically mouse models that people study on certain. So if you want to study certain diseases in wet labs, you either do it through cell lines or you do it through some model systems. So this is one of those model systems is a murine model, and they studied prion disease in that, and uh, basically they did some inbreeding and I think yeah brain transcriptome analysis across multiple time points and inbred strain. Basically you have a control and you have a disease strain, and then you kind of inbreed. You have the first generation, as you'll have basically mixed strains in the first generation where you will have disease and normal urine models. So that's the function of the crossing, basically the inbreeding. And they what they did was they did some brain transcriptomic analysis, basically RNA sequencing, and they identified some differentially expressed genes using statistical analysis, and they integrated this data into the protein-protein interaction networks already known, uh, and basically certain like pathways or certain functionalities came forward from that. So one was glial activation, the other was axon and dendrite degeneration and neural apoptosis. So these are kind of modules that were previously identified, which kind of correlated with the kind of associated with this disease when they mapped out the data onto the network. So uh, these are various types of studies that has been done on several diseases, and the approaches are varying, but the whole idea is to identify certain proteins or certain network of proteins or a set of genes or a set of transcription factors that are crucial to a particular phenotype. Once you identify them, then you can like create drugs to target them. You can try knocking them out. You can do whole lot of things with that. The identification of those important central proteins and genes and transcription factors is very important to identify the cause of the phenotype, which we do through these systems biology techniques. Okay, so next topic we come to is community detection. So this is a very like what to say. This is a very common topic and a very what is a popular topic in graph theory. So if you study graph theory from any CS courses, you'll study these in a more mathematical form. I'll not go into the details of those mathematics, but I'll go into slight details of the algorithms and how they work. But yeah, so what is first to define what is a network community. So basically if you consider your Facebook data, so there's this guy called Eric Belm. In 2011 he was finishing up his PhD at Caltech, and what he did was he displayed social networks to which he belonged in LinkedIn, Facebook, and Twitter. These three networking sites he took, and he kind of mapped out his following list and his friend list as different nodes in the networks. So and he used a visualization tool, and he found that certain nodes came together in a community within the network. So basically these blue points are his grad school classmates. These basically peach color ones are his basically from a grad school club, and uh, these are his, I think, childhood friends and family. So you can see the formation of communities in his these social network, or in anyone's, if you map out your social networks as well, you'll find these kind of communities. So what is a community exactly? So it, it the problem of community detection is basically the problem of finding natural divisions within a network. So like uh, it's not like you forcefully try to make a division. The division has to be natural, and basically uh, the problem of finding natural divisions of a network into groups of vertices, which are called as communities or clusters. So you divide the network into different groups or clusters of nodes, which you called communities. And uh, the property of these communities is that there are many edges within the group and very few edges across the group. So basically, if you think of this example, uh, the childhood friends of this guy are like well connected. So basically, he knows. His friends are well connected among each other, but his friends are not well. His childhood friends are not well connected to his grad school friends. So the the edges within a particular community is much more than the edges between two communities. So this is how you kind of qualitatively define communities. But exactly how what we mean by how many or how few of this linkages between communities, it is kind of debatable, and you can have a variety of like wide range of these connections, and you can call them communities. So there is no hard and fast definition of communities within graphs, but the whole idea is communities within a graph are nodes which are connected very well to each other, but not connected that well to other communities in the network. So you are trying to find set of nodes which work together. That is what community detection is basically is. So where do you use this community detection? In case of biological networks, it is a promising computational approach to discovery of functions of genes and proteins is to identify functional modules in biological networks. So basically, you want to like how our system is designed is that uh, there are certain genes that are responsible for certain phenotypes or certain functions in the cell, and if those functions are related, those genes tend to be connected more. So what happens is modules or communities are sets of genes or proteins that perform biological processes together, and it is possible to classify proteins with unknown function by determining what community do they belong to. So kind of like if you have a protein which you do not know the function to, you can like assume the function of that protein or a gene depending on which community do they belong to. If it belongs to, say, for example, a tumor suppressor gene community, so it's highly possible that that node is also a tumor suppressor gene. But yeah, it is always not true. You need to have certain proofs and certain validations to make this claim. But yeah, you can at least assume. And correct identification of function community is also important in drug design applications, as we talked about targeting hubs and different modules within networks to for drug targets, basically drug designing applications. And communities play a particular role in understanding human diseases that we discussed about. Here is an example of a human disease network. So this is basically, if you see the network, so these are different genes and proteins that are present in the network, and the node size indicates how many other nodes. Basically, it indicates the degree. It is not exactly the degree, but basically, when you move from one, basically the node size goes up, the basically degree increases. So these are not exact degree values, but these are more or less kind of shows you the correlation between the degrees and the size of the nodes. And here are the different classes of those diseases. So if you look in the network a little bit in detail, you see the similar colored nodes are clustered together. So the red are clustered with red, the greens are clustered with green, and the yellows are clustered with yellow. So each of this color represent a particular type of disease, and the fact that the nodes that are related to a particular disease, the genes and in this case the genes that are related to a particular disease are kind of clustered together, kind of forming a community together. So this is kind of the reason that we want to find communities in biological networks, specifically in disease gene networks. So if we if we if we can identify communities which are affecting which are like basically the main reason for causing that phenotype, it becomes easier to identify what kind of interactions are causing that phenotype, and also preparing drug targets for those particular genes and nodes. So the take home message from here is basically all the nodes having same color, meaning associated with the same disease, are clustered or together, or kind of forming a community. Okay. Next, we come to certain algorithms of clustering. First one of them is hierarchical clustering. Before I move into these topics, just to give you a heads up, I have written certain formulas to give you an idea about how these are calculated. But we'll not be going much into the basically the mathematics of it. We'll try to understand how it uh, affects us in the biological system, from a systems biology point of view. But I have I have written down the formulas and how to calculate those distances. If you are interested, you can obviously go and read up on them. If you have any issues, just drop me a mail. I'll be happy to interact outside these TARs. So basically, hierarchical clustering requires two decisions. First is basically the choice of the distance function. So there are a variety of distance functions available. So the most simplest of them is the Euclidean distance, which is basically this squared of the difference in position of two nodes. So how you calculate Euclidean distance in graphs is basically meaning in normal graphs, not node vertices graph. I'm talking about plots. In plots, how you calculate a Euclidean distance, or how you calculate Euclidean distance between two vectors. It's analogous. And we have other distances as well. Also, this is the cosine similarity measure. This is nothing but basically the dot product. So you have a dot b is a cos ab cos theta. So you measure the theta here. So you can see this as a formula. This form is similar to how you determine what angle of two vectors lining up. So basically, you do a dot product and calculate the theta. It's kind of similar here. So you have the squared Euclidean distance, you have cosine similarity measures, you have other measures as well. And one of them is basically the correlation coefficient. So basically, what I talked about the first few slides is how to find similar similar nodes within the network is kind of plotting their degrees as a scatter plot and seeing the correlation coefficient. So this is kind of that approach only. You calculate this Pearson correlation coefficient, and it gives you a measure of how the nodes are like close to each other, or how the nodes are similar to each other. So uh, that is that. So first is the choice of the distance function. You can choose it between any. Obviously, there has to be certain what to say rationale for choosing these distances. You cannot choose arbitrarily. Obviously, you have to give a reason. But yeah, you choose your distance function. And you measure the distance between two nodes in the network, and now you have to see how you measure the distance between two clusters of nodes. So it can be basically, if you imagine two clusters of node, it can be end to end. So basically, you can measure the total. You're measuring the kind of the diameters across the two clusters. So the the farthest node of the left cluster to the farthest node of the right cluster. That's the maximum distance that will be possible between the two clusters. That is one possibility, which is the maximum of this complete linkage clustering. It's basically the maximum of the distance between the two nodes, or you can also calculate the minimum on the single linkage clustering. So basically, uh, the closest nodes in the two clusters, that distance also you can calculate, or you can calculate the distances between the centroids of the clusters, kind of taking how we calculate the centroids, kind of taking the center of mass and determining the center of mass and measuring the distance between those 2 points. So uh, it's basically an iterative algorithm where you like if the distance you measure the distance. If it is above a threshold, you break. If it's below a threshold, you keep it as a community. So those kind of things you need to define before doing hierarchical clustering, but it's a very simple concept. You measure the distance. If it is up above a certain threshold, you divide it into two communities. If it is not, you keep it as a community and you check it with other communities. So it's quite a simple concept, and it's kind of old hierarchical clustering, but it's an effective clustering technique. Is what I can say.
Actually, with like the choice of distance function, like what sort of determines which function to use, or like is it basically the ones that we used before, and then so like is there something like, for example, with the given examples like Euclidean cosine similarity and Pearson
Hmm.
correlation coefficient, is it basically Euclidean was used like it's a basic form of distance, or like is there some metric by which we determine okay this function to be used for this purpose or something like that?
Okay, I'll give you an example. So basically, uh, Euclidean distance has a very nice property. Is the
Yeah.
distance between A and B is equal to the distance between B and A. So
Oh yeah, yeah.
if you are assuming this property, you can go with the Euclidean distance. But if your network has somehow the distance between two nodes, if your network is directed, so if
Yeah,
your
yeah.
network is directed, the distance between A and B and B and A would not be the same.
The same, yeah. Yeah.
So in that case, you cannot use Euclidean distance. You have to think of another distance parameter.
Okay.
So it depends on how you are how you are forming your network and what basically how you are wanting to calculate those distances.
Okay.
The distance function can be chosen by you.
Okay.
Yeah,
Thanks.
yeah. Okay. So moving on is the Garvan-Newman algorithm. So this is an algorithm that is used in basically to dissect the network into communities. So it proceeds in different iterations. So at the beginning of each iteration, you compute the betweenness of the edges in the graph and remove the edges with the largest betweenness. So uh, the metric here that they are calculating on is basically edge betweenness. So if your edge is if your edge has the highest betweenness in the network, you kind of remove that edge, and if more than one edge has the highest betweenness, you remove all those edges at the same time. And basically if you remove the edges, it kind of falls into two communities, and you repeat these iterations, and basically again you go and calculate what is the highest edge between this of a node, and if it is the highest, you cut that off and you do those iterations again. So just to give an example, so here if you consider this whole network from A to, I think, CHI I from A to I. So the highest betweenness centrality is between it is these edges. So F and E and E and A. These two are kind of forming a bridge between the two other clusters in the network. So basically, this if you calculate the betweenness of these edges, this will be the highest. So uh, if once you remove these edges, so like you identify Fe and Ea are the highest betweenness edge betweenness edges, and you remove them, you form three communities. So one is this, next is this, and the third one is obviously E alone. And uh, in the next iteration, you try to remove edges from these networks. So like all the edges, F, G, I, and H, and here A, C, D, and B all have the same betweenness. Because these two, the FE and the EA edges are not there anymore in the second iteration. So all the edges here have the same betweenness. Same is the situation here. So you have to remove all the edges or you don't remove one. So basically, your algorithm is giving you this is one cluster, this is one cluster, and this is the other cluster. So where are you stopping this partitioning is where all the edges have the same betweenness, and if you remove the edges, all will be single nodes. So you do not want that. You want a community. So basically, you remove the highest betweenness edge in the network, and then you form different communities and you look for highest betweenness edge in those particular communities. If all of them have the same highest betweenness, so there's nothing more you can do. That's the last community you'll form. So the algorithm kind of works in this way. So first, uh, uh, you repeat these iterations basically uh, until all the edges are gone, and the partition with the total modularity score is the optimal partition. We'll talk about modularity in a little bit, but basically uh, you can imagine individual nodes to be partitions as well. So here you can see three communities, but this is also, if you remove all the edges, and you can see each node is a community as well. So if depends on how low you want to go, as how granular you want to go into the network. So it's not always like the more granular you go, the more smaller communities you get, it's better. No, it's not the case. So basically you need functional, or you need it's basically you need functional communities. You need communities which which make sense. So uh, you cannot go on like making the communities smaller and smaller and smaller until you need you remain with a node. So how do you determine when to stop this iteration is something through something called modularity. So we'll talk about modularity. So what you do is, if the modularity score is after partition is lesser than the modularity score before partition, you do not partition it. Only if it is higher after partition, you go for the partition. We'll talk about modularity score and what it means, but this is the general idea. So, uh, okay, here comes modularity. So, uh, the first thing to keep in mind is the community structure problem is different. Basically, the community detection problem is different from the graph partitioning problem. The primary difference is that graph partitioning problem, you know the number of elements that you're allowed to have in a in a partition in a graph. But in a community, the number of nodes that will be present in your community can vary. That is not predefined. That you need to find out. Excuse me. Yeah, that is not predefined. It's unconstrained. So you are at liberty to select trivial divisions of the network that puts all the vertices in one or two groups and none in the other, which guarantees we have zero intergroup edges. This division, in a sense, is optimal. As I said, like the more smaller and smaller you make a network, it it like technically it is correct. Okay, the most best community will be a single node itself. Technically it is correct, but from an application point of view, from a biological network point of view, or any kind of network point of view, you do not want that. You want a robust community. So even if more divisions are possible of that community, it's not optimal. So how do you define that? Basically that is what modularity is. But basically the true community structure in a network corresponds to a statistically surprising arrangement of edges and can be quantified using this parameter called modularity. The modularity is basically uh, measures the strength of a community partition by taking into account the degree distribution. So, so imagine you have a network with m edges. The expected number of edges between two nodes I and j with degree di and dj respectively will be di into dj by 2m. So what you measure here is how you measure the strength of the community is Aij will be your basically your adjacency matrix, which gives you the number of edges between ith node and jth node. And dij by 2m will give you the all possibilities of number of nodes between number of edges between two nodes I and j. So it's kind of checking how strong the connections are between the nodes in a community. So you have your original number of edges and minus the number of basically minus the all possible number of edges. This gives you a number which is basically the higher it is, it indicates that it is a good structure. So basically, the higher this value will be, it means the high Aij is higher. Aij higher implies there's more connectivity within the community. So this modularity as a score, we can imagine it as kind of measuring the strength of the community partition and using its degree values. And the higher the modularity score, the better is the community, the more connected is the community. So yeah. So one such optimization problem is just a second. Huh. One such optimization problem is modularity maximization. So you want to have your communities where you have the maximum value of modularity. So uh, the communities that you have should have the maximum possible values of modularity. If it is not the maximum, then you go for another iteration where you divide the network again, so that you can try to increase its modularity. So the intuition behind the idea of modularity is that a community is a structural element within a network that has been formed in a manner far from a random process. So basically, what you're claiming is this is not a random community. This community is present because of certain reasons, and that certain reasons may be functional reasons, that certain reasons may be disease or contributing to a phenotype, but it is not random. There is a reason behind the formation of that community, and it is not random. And that is the whole idea of what modularity measures. So if we consider the actual density of links in a community, it should be significantly higher than the density that we would expect if the links in the network were found by a random process. So basically, uh, if you think of a null model, we talked about ER models, ER networks, and WS networks last time. So if you think of a null model where you're making a randomized network, and the idea of the community of a community in a network is basically uh, in the randomized network, the density of the nodes within density of the edges within those nodes in this community will be much lower. So in your original network, those nodes will be much higher will have much higher connectivity or high degree centrality compared to the nodes that has been and the nodes in the random network. So what you're claiming is this community has not formed by chance. There's a statistical significance associated. This is not a random process. So basically, in other words, if two nodes I and J are ending up being in the same community, there should be more likely a link between them. So basically, Aij will be one. So your modularity score, if you see the formula, if your Aij is one, basically if you can consider uni connected nodes, so multiple nodes are not possible. So Aij can either multiple connections are not possible. Sorry, pardon me. So multiple edges are not possible between nodes. So Aij will be either zero or one. So if Aij is one, your strength of community will be greater than than the next alternative is zero. So if your Aij is one, it will lead to a more higher overall value for the modularity. And if I and J end up being in a community such that the chances of having a link between them is the same as between any two nodes in the randomized network, then the Q value is more likely to be low because there will be uh, mm, certain pair of nodes I and J where Aij will be 0, and that will bring down the value of q. It's basically you'll take an average of the modularity between the nodes in the network. So in this case, if it is connected, you'll always have a value of 1. And if it is basically the chances of connection is the same as the random networks. So in some cases you'll have 1, in some cases you'll have 0. But ultimately it will be less than if it is connected. So Aij will be 1 only if it is connected. And if it is not connected, it will be some value less than 1. But yeah, it will not be 1, because you'll have in a randomized network you'll have those nodes in like you'll randomize it iteration after iteration, no? So in some iterations you'll find uh, those nodes that are uh, those nodes are not connected. Basically Aij will be 0. That will bring down your value of q. So this is the whole idea of modularity maximization. So we'll take an example from this. So we'll try to implement the Gardner-Newman uh, algorithm to this graph with the optimal optimal communities being defined as like the communities with highest modularity values. So this is a graph with 15 edges, as you can see. 15 nodes, 15 or 14, I think. Yeah, I think it's 14. Yeah, 14 nodes. So if you are given this graph and you are told to implement the Garvan-Newman algorithm with the constraint that you stop the partitioning when the maximum modularity has been reached. So let's see. So you calculate basically first you see which has the highest betweenness edge in this whole network. So the highest betweenness edge will be this 7, between 7 and 8, because these two nodes and the edge is kind of acting as a bridge between the two sides of the network. So you remove this edge from 7 and 8, and then you calculate the modularity. So modularity of 1, 2, 3, 4, 5, 6, 7. So 1, 2, 3, 4, 5, 6, 7. So this is one community you'll calculate. And you calculate also the modularity for this community. So 8, 9, 10, 11, 12, 13, 14. I think it's given till 17. This will be till 14. So it's basically you calculate the modularity scores, and you combine those to get a total modularity score. So you have two communities here, and you calculate the modularity for each community, and you add them up to get the total modularity score. This is for the first iteration. Now, if we do the second iteration, the highest betweenness edges will be these between 3 and 7, 6 and 7, and also 8 and 9, and 8 and 12, because these are now acting as bridges between these cliques. So these will be the highest betweenness edges. So if you remove them, you calculate a modularity for, you have now 1, 2, 3, 4, 5, 6 total communities. But basically these are basically single nodes. So basically you have to calculate the modularity for these four. So you calculate the modularity and it comes out to be 10.12. So if you now try to like go for iteration 3, because this will be the ideal partition. But even if you are not satisfied with that, if you want to go to for iteration 3 and you break open these, or like these will have the same betweenness value, no? So if you try to like break these edges, all the edges will be broken. Sorry, all the edges will be broken. So you'll be left with individual nodes. So because these have all the same edge betweenness. So 1, 2, 3, the edges between them will have the same betweenness value as 4, 5, 6, or 8, 9, 9, 10, 11, or 12, 13, 14. So one more iteration into into partitioning of the community, you will be left with only nodes. And the community, if you calculate the modularity value, that will be much lower than the 10.12 that you have here. So uh, basically, uh, uh, much lower, what I'm saying is basically it will be 0, because it will not be connected at all. So uh, this is the highest modularity score you will get. So basically this will be your optimal partition. So the final partition will look something like this, with 6 clusters. So each triangle on each side will be a cluster, and these nodes will be a clusters of themselves. So this was an implementation of the Galvan-Newman algorithm where you, um, I'll go through it once again. So first you calculate the highest between these edges and you remove them. So in this case, it's 7 and 8. You remove that, then you are left with these two whole clusters. Now you see the highest betweenness edge between in these clusters. This will be between 3 and 7 and 6 and 7. Here it will 8 and 9 and 8 and 12. Now you remove that and calculate the total modularity. You will get your partition looks something like this. And now all the communities, if you try to go for another iteration, move another edge that has the highest betweenness value, will be left with single nodes. So this will be your optimal partition with your highest modularity score. So these kind of algorithms actually are implemented when you are trying to when you are doing community detection. So uh, your ideal uh, your ideal algorithm will be you'll basically find the communities with the highest modularity values, and uh, yeah, in every iteration you'll calculate that modularity value, and if in the next iteration if the modularity goes down, you will not make that partition. The previous one will be your final partition. So this is an overview of how the Galvan-Newman algorithm actually works uh, in basically networks. So yeah, you can try this. I think in NetworkX in Python, you can try this. Obviously, this is available in MATLAB as well. I'm a little bit more familiar with Python, so I suggest Python. But yeah, obviously, if you're comfortable with MATLAB, also your course is based on MATLAB, so you should focus on MATLAB. But yeah, if you want to build more play around with some stuff, network X is a very good library to do that. So yeah, you can implement these algorithms, this hierarchical clustering, Galvan-Newman, and other algorithms as well. Okay, so yeah, this is the final uh, topic for our discussion today. After this, we'll try to solve some questions. So the final topic is network motives. So what are network motifs, or what are motifs in a network? So the motifs are basically subgraphs that are overrepresented compared to a randomized version of the same network. So it's kind of the similar idea. If you generate a random network, is like basically you identify a subgraph in your old network, and you generate a random network, and you see if that subgraph is getting formed in your random network. Not like one iteration of random network. Say you do say 1,000 iterations of a random network, and you see your subgraph that you identified from your own network is getting formed only one or two times. So you can say it's the mm, the subgraph that you have in your own network that is not random. That is not possible through random chance. That is there for a reason. So that has some functionality together, or some characteristics together. That's why they are together, together as a subgraph in your network, and not getting formed in your randomized network. So that is the whole idea. So the motifs are basically subgraphs that are overrepresented compared to a randomized version of the same network. To identify motifs, first is you have to identify all subgraphs with of n nodes in your network. Then you randomize that network while keeping this is very important, while keeping the same number of nodes, edges, and degree distribution. So basically your degrees distribution, your nodes and edges, basically all the essential parameters of your network should be same. That should not be different. And then you randomize that network and see if your subgraph of your original network is getting produced in this in the randomized network. And you identify the subgraphs of n nodes in the randomized version, and the subgraphs that occur significantly more frequently in the real network as compared to the randomized one, they are designated as motifs. Basically, uh, you're claiming that this pattern of subgraphs is not random. These are together for reason. That is what you're claiming. When you're claiming when you're claiming a subgraph to be a motif of a network. So you're saying is this is not random. This is here for a reason. And like kind of to to generate these randomized networks and to identify these network motifs, here are two models I have given. So there can be multiple others. These are two examples that was, I think, talked about in the videos, but still, if not, also I'll talk about them. So first is the configuration model. It generates a random graph with arbitrary sequence of degrees. So uh, you can have like an arbitrary sequence of degrees in a node, and uh, what you do is basically, uh, if you cut a node, if you have a as a node connected to a node, you cut an edge through the middle. So what you'll be left with is basically nodes and half stubs of the edges. So you'll have something like this. So the previous degree of this node was 3. Now if you cut the stubs, you basically have half edges over each node, and uh, what you are keeping constant here, if you look carefully, is the degree of the node. You will not be changing the degree of the node because those half stubs are there. So after that, what you'll do is you'll randomly connect any two half stubs with each other the with the constraint that like kind of no stubs should remain. So like basically you say you have a network of 3 or 4 nodes, and you make these stubs and you connect them, and after you connect you find that there is one node left. That is not that will not be ideal. So you have to connect all the nodes with each other with those half stubs, and basically repeat until there are no stubs remaining. What is the limitation of this model is that there can be a possibility of self edge or a multiple edges between the same nodes. So for example, you have 4 nodes and you cut the stubs, and say for example, you have connected the 3 nodes together, and one is separate, and you could not connect it to any one of the 3, and it has 2 stubs. So if you join those 2 stubs, it will form a self edge. So so you when you are implementing this model in your program, you have to keep in mind of these constraints. So like there is a high possibility that it can implement a self edge to itself, or it is highly possible that it can form multiple edges between the same number of nodes. So these things you have to keep in mind when you are implementing the configuration module for random networks. Another model is basically edge switching model. So you randomly select a pair of edges and you rewire them, and you repeat this process of rewiring until a random version of the network is produced. So if you do this rewiring say once or twice, so that will not be a random network from the fact that your original network and the network that you have formed by changing one or two wires, that is kind of a close sister to the original network that is present. So that will not be that you cannot classify as a random network. So you need to like pass it through a number of iterations, and you need to rewire these edges between these nodes randomly, and you repeat this process until you have uh, a random network. So until you have basically a network uh, which is quite dissimilar to the one that you originally had. So this is what the edge switching model process. Just take a random pair of edges and then you rewire them, and you go on doing this until you form a quite a different version of a network from your original one, and then you can compare it if it has that motif or not. Like for finding network motifs, basically your randomized network can form that subgraph that you are looking for, that you are aiming to be a motif in your original network. So yeah.
Actually, for configuration model, so there's this definite endpoint, right? Where you
Huh.
repeat the iteration, still there are no stubs remaining essentially.
Yeah.
But
Yeah.
with switching, there's this vague endpoint where you sort of,
Yeah. Yeah.
so like, is it basically we sort of do the same thing many times and then sort of take, how do we sort of determine
So
like
it's basically
if it's different
determined
or
by
not?
It's basically determined by the number of iterations you are going on for this rewiring process. So
Mm.
it depends uh, uh, on the number of edges in your network. So if your network
Okay.
has say 500 edges, you are rewiring
Yeah.
it for like one or two times. It
Oh yeah, it
doesn't
doesn't.
qualify as a random network, does it? But if your
Yeah.
network has a 500 edges, you are rewiring for 500 times. That can
Mm.
qualify as a random network.
Okay.
Right?
Yeah.
It depends on the network you have.
So even there's no like arbitrary, like there's no definite
No, there's
rule
no
per se.
no no no.
Yeah.
There's
So
no
it's
definite rule.
yeah.
You it's arbitrary, mostly
Yeah.
arbitrary.
Yeah. Okay. Yeah.
Okay. We'll move into questions. Uh, this was all the topics that were covered. I think I've covered all of them, but yeah. We'll do some MCQs now. Start off with the first one. I'll come back in 2 minutes. I'll be back.
Yeah.
Start
Sure.
doing
Sure.
it. Yeah.
Yeah. Yeah. So I think it's only two.
Oh, okay. Oh, Akshay, you are here alone only, so yeah, you can give me the answers.
Yeah. I
So
think yeah, it's only two.
it's
The
only
second.
no, it's not.
But
Basically, so
no. Sorry. Yeah.
so if you want to say, for example, when you are comparing motifs, so if you are trying to find motifs in a network,
Yeah.
so your idea was the whole idea is to keep the same degree of the nodes, to see keep the same number of nodes and
Yeah.
keep the same number of edges basically,
Yeah.
and then form a randomized network. So
Yeah.
a fixed number of edges connected randomly by n nodes will be a random network, right? So your number of edges are same, you're just dividing them.
Okay, 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 okay. Yeah, 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 yeah. Okay. Now, for
Yeah.
me, the confusion was with this fact that okay, I was wondering, okay, the nodes would be fixed, and then but then the number of edges would change, and
Huh,
the degree
huh.
might also change. Yeah, yeah.
Yeah, yeah. Okay. Okay. Moving on. Give an explanation also, like true or false.
I feel like it will be false because I mean it is essentially a fraction. Like clustering coefficient would be essentially a fraction of.
Sorry, I couldn't hear you.
Yeah, so like, I feel like it will be like dependent on its degree in a way because it's essentially the fraction of what
Hmm.
like yeah the fraction of edges that are connected. We are sort of calculating the fraction of edges that are connected, fraction of nodes, neighboring adjacent nodes that are connected.
Yeah, it's true. It's true. Yeah, yeah, yeah. I got your answer. Like
Yeah,
in total fraction, it's the like you need the total number of
yeah, yeah,
degree, basically the degree of the node to calculate the clustering coefficient. Yeah.
yeah, yeah.
Okay. Next.
So if network is disassociative,
Hmm.
essentially there is lesser chance of like there is less tendency for the nodes
Hmm.
connect to other nodes with the same properties. So
Yeah,
essentially, yeah, so it will be less resilient.
it will be less resilient.
Yeah,
Why? Like okay, okay. Acha, I got it.
yeah.
Yeah.
Oh wait, yeah, yeah, yeah,
Yeah, it's the same. But it's basically if it is disassociated, yeah,
yeah,
the node. If you have two communities, the node in community one will be connected more to community two than community one than
yeah, yeah,
nodes in community one. So
yeah,
if your nodes are not connected to the individual community, that means in that individual community on module, the functional importance of the node is very low, or
yeah, yeah.
basically the participation of the node is very low. So
Mm.
if you target that,
Then it will be more vulnerable.
it'll not affect that module that much compared to the
Uh,
one compared to a one if it is associative.
okay, okay, okay. Oh, okay, okay, okay. Okay.
This is a multiple answer.
Yeah, yeah. And what do you mean by special nodes?
Special as in I'm talking about hubs or
Yeah,
uh,
yeah, okay.
like yeah, important nodes in the.
Yeah. Mhm. Yeah, so there will not be any special nodes. And then, yeah, the distribution is uniform for P equal to 1, and any two nodes are equally likely to be connected.
Hmm, correct. It's first, second, and third. Yeah,
Yes.
good. Nodes and edges.
3.
Three. Okay. Yeah. It's basically motif identification is basically the subgraphs, like
Yes.
identifying a particular subgraph in a network that is more significant than uh, more
Okay.
significantly formed in a network compared to a random network. So
Isn't it the same as like motive identification essentially? Or
sorry,
it,
I couldn't hear you.
no, would it be C?
I think the answer is C. Yeah. Yeah, it's basically motif identification. There's one, this option is a little bit tricky,
Yeah,
cliques of high degree nodes, but the
yeah,
point is the cliques will be like the smallest triangles, like
yeah, yeah,
A, B, C. A is influencing B and C, B and C are influencing each
yeah,
other. So that is kind of, that is like a dense connection, but the point is you cannot uh, basically mm, uh, that will be the smallest dense connection, right? The clique will
yeah,
be
yeah,
the
yeah, yeah,
smallest.
yeah.
It will be the triangle. So it is technically a denser connection, but the motif will be a much denser connection than a
Yeah,
single
yeah.
node.
Zero.
That's true. If complete graph will have
Yeah, no,
no
notice.
nodes.
I mean, am I supposed to select the correct answer options from the?
Sorry.
Am I supposed to select the correct answers or which, the two statements?
Yeah, yeah.
C and D are true, I think.
Okay. The answer is C. I'm not sure D is for connected to all.
One is connected to four, two is connected to four, four is connected to
Uh,
three, and
this is
five
a
is connected.
this is by the way directed graph if you notice. So
Yeah,
two,
yeah.
so four is connected to three, but three is not connected to four.
Ah, okay, okay. Both
Uh,
ways, yeah.
yeah. So yeah, this is a directed graph.
Hmm.
This is basically just to give you remove node is basically does what the name suggests. It removes
Yeah,
all
yeah.
the nodes there. Okay. Yeah. This is basically a network implementation.
Two. I mean B.
Hmm. Yeah. So that was it.
Yeah.
That's all for this week. Before you go, since we have time and there is no one else in the call, I just wanted to know if you have any suggestions for me to how to conduct these classes because as I don't know if you know, this is also my first live session. So this is the first time I'm taking this kind of live sessions. So it's new for me too. I would like to know if you have any feedback or any suggestions regarding how to conduct these and like if you can do anything more else or something that would help you.
Yeah. For me, actually, this system works really well because after listening to the lectures, essentially, this is
Hmm.
more like a recap session where some of the concepts are sort of like I understand it better because like I've gone through the lectures once and
Hmm.
then read materials on my own, and then in this session, basically, when you're going through the lectures again, so I sort of it's more like a revision session where oh, I understand some concepts better, and then you provide some additional info also, which keeps it engaging. And then the system where like the initial an hour or like an hour and quarter essentially is for revision, and then you go to question answer sessions where
Hmm.
so we have gone through a complete revision and then we are sort of applying them. Works really well for me. I don't know about the others, but yeah, for me this system works really well. Yeah. So
Okay. And how are your assignments and everything going on? Like, are they
yeah,
good?
I mean, yeah, they're going fine. I mean, weekly assignments like more or less by the time the weekly lectures are done, it's easier to sort of go through the assignments. I mean, like for like finish the assignments quickly.
Oh,
But then the thing is like with the non-graded assignments, there's always this chance of like you get this immediate feedback, right? Whether okay, this answer is right or wrong, or like what
hmm.
went wrong, things like that. For the graded assignments, it takes some time, right? After the deadline is done and things like that. So
Okay.
like it's more or less, yeah. I complete it and then so wait for some time in order for the feedback to come and things like that. So, but it works well. I would say.
Okay. Okay. Just to like I had asked you this in the first day. I just forgot. What is your background?
So I finished my BSMS in ICT Trivandrum.
Okay. Okay. Acha.
Yeah,
And your
uh,
major was in?
biology.
Oh, okay. So you have taken courses like this before? Like
Not quite. I mean, we had this bioinformatics course, but then it was basic, like introduction to some algorithms, like
blast and stuff, right?
blast and things like yeah.
Yeah.
So
Yeah. Okay.
nothing,
Okay.
nothing. And then even programming also it was more like yeah, basic R code, like just define a variable, things like that. But then
Okay.
like in other than that, I have been like sort of trying to learn programming code like Python and R on my own. But then it's more like it's basically theory or like you're just studying from scratch. It's not related to any project or like applying it somewhere.
Oh,
So
okay.
yeah,
I'll suggest like there are if you go to Kaggle, there are a lot of data available. There are a lot of competitions available.
oh, okay.
A lot of stuff you can do with data. So I suggest you visit Kaggle and uh, you are aware of databases like TCGA and stuff
Yeah,
like
yeah,
that,
yeah.
right? So data is there is bhar bhar ke data. Like
Uh,
and the amount of data that is there is huge. You just need to pick up anything and start analyzing.
okay,
That's
okay,
what
nice.
you need to do. Yeah.
But
So
then, yeah, this sort of like, and I was sort of working in Norway for a couple of years, like on a
okay.
project. Yeah,
Okay,
I just
okay,
came back and then yeah,
okay, okay.
we're going
Hope you're enjoying this
yeah,
course.
yeah,
Yeah,
it's fun. Yeah, thank you.
thank you. I'll
Have,
close this course,
yeah,
close this session today. Yeah.
yeah, thank you.
Thank you. Thank you. See you next week.